Hello, my name is Harmit Malik. I have the honor of being the current president of SMBE and one of the main organizers for SMBE 2021. I want to welcome you all to this experiment in the virtual meeting. I want to thank the three other people who were my primary collaborators in this process. Beth Shapiro from University of California, Santa Cruz, Mary O'Connell, who's from the University of Nottingham, and Lulu Stader, who's our executive administrator of the society. I also want to give a special thanks to the rest of the SMBE Council and to Chris Lapine from Allen Press. In addition to these individuals, we were helped a lot by hub organizers across 3 different time zones who coordinated all our efforts along with a lot of different symposium organizers. I'll get a chance to thank them in more detail and give you some additional insights into how we organize this meeting at the SMBE business meeting. So I'd encourage you all to tune in for that description. Today's talk is going to focus on work in my lab that focuses on antiviral proteins and how we can use mutational analyses in the lab to identify constraints that shape their adaptive potential. This is work that was done by Jeanette Tentere, who's a current postdoc in the lab, Rosanna Colon-Tije, who was a former graduate student in the lab, and both of them were co-mentored by Michael Emmerman, who's my virology colleague at the Fred Hutchinson Cancer Research Center, where all of this work was performed. My lab is very interested in genetic conflicts. This concept derives from the fictional character, the Red Queen, who was introduced to us by Lewis Carroll in his books. And this is a very memorable line in the book Through the Looking Glass, in which, in response to Alice's complaint that they've been walking for a long time and haven't really gotten anywhere, the Red Queen remarks that in Wonderland, it takes all the running you can do to keep in the same place. This was seized upon as a really important way to synthesize a number of ideas that had been percolating in evolutionary biology, most prominently by Lee Van Valen and his peers, as the Red Queen hypothesis, who pointed out that in systems of competition, for example, prey-predator interactions between snow leopard and snow hare populations, it was not just enough to adapt to the abiotic environment for these organisms. They also needed to keep pace with the adaptive strategies of the competing species in that fitness space. And if they stopped evolving, they ran the risk of being driven to extinction by virtue of the relentless nature of this competition. We are very interested in taking this ecologically inspired approach and applying it in molecular terms, taking advantage of molecular evolution analysis. And in particular, we are interested in studying genetic conflicts that occur between different genetic entities, for example, host versus viruses, which is going to be the focus of my talk today, or even competition within the genome, for example, transposable elements and the rest of the genome, or centromeres and centromeric proteins, which is also another focus in the lab today. We describe these as extrinsic versus intrinsic competition. Returning to this competition between antiviral proteins and viral proteins, particularly for pathogenic viruses, antiviral proteins impose a significant challenge to the success of viruses. So they can engage with viral proteins upon entry into a cell by recognizing specific features of the viruses, and by virtue of that recognition can extinguish the virus first within the cell and then eventually from the species. This imposes a lot of selective pressure on viral populations to explore new states such that they can no longer be recognized by the existing versions of the antiviral proteins. Over evolutionary time, this will force host proteins to also adapt to now recognize the newly evolved form of the viruses. I hope you can recognize that the viruses have a tremendous advantage here in terms of their adaptive potential, both in terms of mutation rates and effective population sizes. But it's not enough for the viruses to just escape one antiviral protein. Most mammals encode nearly 200 antiviral proteins that are dedicated to the task of engaging with and extinguishing different viruses. And so for the host to be successful, it really needs to win one of these arms races. Whereas for the virus to win, it needs to successfully run through the entire gauntlet of the antiviral proteins that are presented. And that's where the math begins to even out a little bit. Even though this again seems like a many-to-one interaction, it's actually not, because any given antiviral protein is not just dealing with one pathogenic viral species, but instead a whole plethora of them that are encountered over the course of an organism's lifetime. So you can see that this is a many-to-many interaction, but even if we were to simplify it to a one-to-one interaction, you can appreciate that this is actually an engine for relentless evolution, very much like the prey-predator interactions. And what drives this engine is the fact that one party is always losing this arms race. Therefore, there's always going to be an evolutionary advantage to be gained by innovation. And we study this innovation by looking through backwards through the history of host and viral proteins to identify signatures of adaptive evolution that have been trapped within these sequences. So in these high-stakes games for binding affinity, where either the host and viral proteins will interact and the host will win, or the virus will escape from this binding affinity, what sort of shapes these back and forth interactions are often amino acid mutations directly at the interaction interface. Therefore, we wondered whether we could use positive selection as a means to identify specificity domains or host-virus interaction surfaces, even in the absence of any biochemical knowledge of what these interaction interfaces were. In other words, by asking where positive selection occurs on the surface of these proteins, could we actually infer not just the biochemistry of the interaction, but also susceptibility determinants, or why one species is much more susceptible to a particular pathogenic virus, whereas another closely related species is not. It actually turns out that even though we have very similar immune repertoires as those of many other simian primates, each of our immune proteins has been tuned to a different binding specificity because of a different history of interactions with pathogenic viruses. And this has turned out to be a very successful strategy to identify these interaction interfaces and susceptibility determinants for a completely different variety of antiviral proteins. You can see here we have completely different unrelated mechanisms of antiviral proteins, including TRIM5 alpha and MXA, which I've bolded here because that's going to be the focus of my talk today. And this is in spite of the fact that they actually work with different enzymatic mechanisms against completely unrelated types of viruses. But what they all have in common is that these games for binding affinity are dictating the ultimate consequence of the interaction, whether the virus will win or the host will win. In today's lecture, I'd like to focus on understanding what are the constraints and the adaptive potential that we can learn specifically within these rapidly evolving domains that define the specificity of antiviral proteins. So in the first vignette, I'd like to present TRIM5 alpha, which is a very important antiviral protein that was discovered by Joseph Zadrowski's lab. And it was discovered because rhesus macaques are almost completely immune to infection by HIV. And the reason they are immune is because after the viral capsid enters the cell, the rhesus TRIM5 alpha is able to form this very rapid lattice-like sheet around the incoming viral capsid and rapidly degrade it before the retrovirus has any chance to go on with the rest of its life cycle and integrate into the genome. Because this is a very early intervention, this can be almost a complete cure, if you will, to protect rhesus macaques from being infected with HIV-1. We also have a TRIM5 protein that is expressed in most cells in our body, but our TRIM5 protein is unfortunately not tuned with the right specificity. Therefore, we do not actually get a significant biological protection against lentiviruses such as HIV-1. Using the same evolutionary approach, we basically identified a very small patch called the V1 or variable loop 1 patch that has rapidly evolved in TRIM5 alpha across simian primates. It turns out that most of the positive selection can be identified as occurring in this very small patch of residues that I've highlighted for you here as a result of these back and forth arms races for binding affinity. In particular, there are 6 amino acid positions, as you can see here, that are very, very different across primates and also differ between human and rhesus TRIM5 alpha. Keep in mind that human has very poor protection of HIV-1, a 2-fold protection, whereas rhesus has nearly 100-fold protection, which is biologically protective. Interestingly, just swapping in this rapidly evolving patch from rhesus into the human TRIM5 alpha backbone is sufficient to confer significant gain of function protection to the otherwise ineffective human TRIM5 protein. And even just a single amino acid residue position change from an arginine to a proline at position 332, as I've highlighted here, is sufficient to give you nearly 15-fold higher protection that is sufficient to actually, again, confer biological protection against HIV-1. In light of the fact that even single residue changes at these binding interaction surfaces can have such profound consequences, we began to wonder what the landscapes of binding affinity may look like for these host-virus interactions. There are two alternatives here. Alternative number one, which I've cartooned on the left, are these sort of rare cliff-like motifs where actually it's really difficult to get the perfect lock and key mechanism to ensure binding, which means that as you take any kind of walk around this mutational space, it's very easy to fall off this cliff of binding and therefore lose your specificity of interacting with a particular ligand. In fact, deep mutational scan analysis of a number of ligand interaction domains have identified these rare cliff-like domains actually are pervasive in host-host interactions, where it's quite easy to lose this interaction interface if you make mutations specifically at the interaction domain. We speculated, however, that antiviral proteins might not be evolving under the same regime, because if that were the case, it would be very, very easy for the virus to escape this binding affinity, leaving it an ineffective strategy on the part of the host. Therefore, we wondered whether they might be more of a rolling hill landscape rather than these rare cliff black diamond ski slopes. To try to differentiate between these possibilities, we decided to take again a deep mutational scan approach, but this time focused entirely on the V1 loop of TRIM5 alpha. So for this, we basically mutagenized all of the positions in the V1 loop, one residue at a time, sort of going through all 20 amino acids at each of these positions. We introduced this library of TRIM5 variants into cat renal fibroblast cells. Cat cells are natively devoid of TRIM5 alpha because they've lost TRIM5 alpha at some point in their evolutionary history. Therefore, all of the TRIM5 variants encoded by this approach basically introduces the single variant of human TRIM5 alpha into cat cells, which we could then select for by puromycin selection. This gives us a library of cat cells, each expressing a different TRIM5 variant, which we now expose to an HIV-1 GFP virus. So this virus is capable of only one round of infection, and upon infection will basically integrate GFP into the host genome, because that's the only part of his genome that's actually left intact. As a result, any cells that turn green are presumed to be not capable of preventing HIV-1 infection. So we refer to these as non-restrictor variants, where any cells that remain non-green either encode a restrictor variant of TRIM5 or in fact encode were lucky enough to not actually be infected with HIV-1 in the first place, in spite of the fact that we do these experiments at a high MOI or a high multiplicity of infection. So to rule out between these restrictor versus uninfected possibilities, we basically sorted the GFP negative cells and subjected them to a second round of HIV-1 GFP infection. And now any cells that remain non-green are much more likely to basically encode restrictor versions of TRIM5. Recall that human TRIM5 starts off being a non-restrictor. So what we're looking for is a gain-of-function mutation that would allow the human TRIM5 variants to now restrict HIV-1. And by deep sequencing the starting and the ending library, we hope to basically enrich for these variants and identify how much they are enriched over the wild-type human. Nicely, each of the biological replicates that we did beautifully correlate with each other. And we were also able to do a couple of other sanity checks, including in this waterfall plot shown here, an enrichment in the restrictor pool. So for example, many synonymous variants that we introduced as part of our PCR mutagenesis strategy are shown here in blue. They actually very nicely co-segregate together. Keep in mind that this is on the right-hand side of the graph because wild-type human TRIM5 alpha is actually quite poor at restriction. And then stop codons are at the very right end because they're actually truncation versions of TRIM5, which would be completely ineffective at restricting HIV-1. Taking advantage of this sort of variance, we also then individually tested in variants that were super enriched for gain-of-function restriction against HIV-1. And to do that, we singly cloned each of these variants into the same cat cells, but this time actually did a dose-response curve to identify how much virus needed to be introduced in order to infect 10% of the cells. So you can see here that in the absence of TRIM5 alpha, we basically get to 10% very quickly. In the presence of human TRIM5 alpha, this is slightly moved to the right because human TRIM5 alpha does have some protection, even though it's not biologically protective. And similarly, we were able to test each of the variants that we discovered as part of our deep mutational scan to identify how much of a protection that they actually were able to confer. And what's really nice is that these individually tested restrictor variants actually beautifully correlated with the enrichment scores we found in our pooled assay, showing that, in fact, even though we had tested all of the TRIM5 variants all at once, we were, in fact, able to get meaningful restriction. Interestingly, 57% of all single point mutations in the V1 loop were able to confer a gain of function in terms of HIV1 restriction, and some of the very best versions were at least 15-fold more potent, which would be very biologically protective. Why is it that it's so easy to gain function for HIV1 restriction in human TRIM5 alpha? It actually turns out that the primary reason is that we discovered that the overall charge of the V1 loop was actually the key determinant of dictating restriction potential. So as you got more and more negatively charged in the V1 loop, you gain more and more of a protection, suggesting that what limits the TRIM5 restriction of HIV1 capsid or interaction with the capsid is a charge repulsion. As you get rid of this positive charge, you're much more likely to interact with and therefore restrict the HIV1 capsid. So it actually turns out that useful mutations in the human TRIM5 alpha are not rare at all. In fact, these gain of restriction mutations are quite common. So then we turned our attention to it's easy to gain function, but is it also equally easy to lose function in these antiviral proteins? To identify loss of function mutations, we again repeated the same scan, except this time we started with rhesus TRIM5 alpha, which actually has significant protection against HIV1. And now we scan to screen for loss of function variants in the rhesus TRIM5 backbone. So variants which, upon mutation, would now turn green in spite of the fact that rhesus TRIM5 alpha itself, the wild type version, would be non-green. Once again, we were really surprised to find that most mutations in the V1 loop actually had no effect on restriction. So you can see there's a significant difference between stop codon mutations and wild-type rhesus TRIM5, which is very effective. And yet, when we enriched for the non-restrictor pool, we found that most of the missense mutations were very far to the right, which means they were all still capable of restriction. This is not just true of rhesus TRIM5 alpha. It turns out that when we are able to confer gain of function protection to human TRIM5 alpha with a single point mutation, the arginine to proline mutation that I already described to you, now this variant version, which we've engineered in the lab, is also very, very resilient to mutation in terms of single amino acid mutations are not likely to lose this newly gained restriction. So not only is it easy to gain restriction, once this restriction is gained, it's actually not so easy to lose this restriction potential. This is not just true against HIV1. We discovered that rhesus TRIM5 alpha as well as human TRIM5 alpha are also quite resilient against other retroviruses, such as this entropic murine leukemia virus that I've described here. So the really surprising result is that the antiviral proteins such as TRIM5 alpha appear to operate in an easy to make but hard to break paradigm, where most mutations do not break restriction and novel restriction is quite easy to gain by mutation. This might, of course, be very important for antiviral proteins to keep pace with very rapidly evolving pathogenic viruses such as HIV1. The biochemical underpinnings of this may lie with the fact that the V1 loop of TRIM5 alpha is highly disordered. So shown here is sort of an artist's rendition of an ensemble of different states that the V1 loop can employ, and this might be the key to the evolutionary flexibility that is able to display in terms of its mutational resilience. This is highly reminiscent of the disordered sort of loops and antigen recognition sites of antibodies, which allow it to recognize and keep pace with changing antigens, despite the fact that they're all anchored in a very structurally stable backbone. This is not just true of TRIM5 alpha. We've actually seen this theme of disordered loops also show up in other antiviral proteins, including the MXA antiviral protein, in which most of the positive selection appears to be concentrated in this disordered loop domain called the loop L4 domain, which I'll tell you about on the next slide. The MXA antiviral protein works on a number of viruses, including influenza-like orthomyxoviruses. And previous work by a former graduate student, Patrick Mitchell, had shown that just like the V1 loop of TRIM5 alpha, it is this unstructured loop L4 domain of MXA that's the primary recognition determinant of the nucleoproteins of influenza-like proteins. That is at the heart of MXA restriction of influenza-like viruses. MXA is a very important firewall for influenza-like viruses, which prevents most of the bird-adapted flu viruses from jumping into the human species. In fact, most of the flu viruses that have managed to gain pandemic status in humans have acquired the same types of mutations in the NP protein to evade human MXA, just highlighting the importance of these kinds of firewall proteins such as MXA. So Patrick had actually focused on the loop L4, again, primarily driven by this adaptive signature of positive selection that he had seen. So he had shown that in the absence of MXA, there is actually ample restriction of an influenza-like protein called tegodovirus, which is actually a tick-borne rodent virus that is actually ineffective at infecting human cells. And the reason it's ineffective is because human MXA is a very potent restrictor of tegodovirus, whereas African green monkey is not a restrictor at all. Yet, if we just swap in the loop L4 domain of human MXA into African green monkey, you can see significant gain of function protection against tegodovirus. And even just a single amino acid residue at position 561, a change from a valine to a phenylalanine is sufficient to confer this gain of function protection. Again, the themes are very similar to what I've described to you for TRIM5 alpha. It is this rapidly evolving domain, the loop L4 domain, that mediates the target recognition, and single residue changes can drastically alter the antiviral properties. In both the TRIM5 alpha and MXA cases, what we've been able to do is look backwards, looking through the evolutionary history of these proteins across simian primates to identify the specificity domains. But what I'd like to do in the last few minutes of my talk today is say that how we can take this evolutionary signature to potentially devise new strategies to understand the adaptive potential, potentially with the goal of yielding potent specific designer antivirals against particular viruses of interest. So for this approach, Rosanna once again returned to Patrick's work that had identified loop L4 as the primary determinant for restricting influenza-like viruses. And instead of doing a single residue deep epithelial scan of loop L4, she decided to actually combinatorially mutagenize all 5 rapidly evolving positions in loop L4, hypothesizing that within this 20 to the 5th domain space, she might actually uncover additional antiviral proteins, perhaps with even more potency against tegodovirus than we had previously identified. This is in light of the findings from ours and other labs that human MXA is actually the most potent restrictor of tegodoviruses. So when she did her first screen, she quickly recognized that the residue at position 561 absolutely needed to be an hydrophobic aromatic residue, such as a phenylalanine, a tyrosine, or a tryptophan. So Rosanna performed a second screen in which she fixed a residue at 561 as a phenylalanine and only allowed 4 positions to mutate in combinatorial space. When she did that, she realized that about 65% of the residues were as good as human MXA, or even better, whereas 35% of the variants were actually worse than, even though we had actually only messed with rapidly evolving positions of loop L4, there was still a lot of constraint acting upon these positions. Of course, we were very encouraged by the fact that towards the right-hand side of this waterfall plot were variants that was significantly better than wild-type human MXA at restriction against the tegodovirus. And that's also shown here on this slide where we individually retested each of these variants. So the first graph shows T103A, which is the catalytically dead version of MXA that loses all restriction. We next have the wild-type human MXA shown here in blue. Keep in mind that this is the best natural restrictor of tegodovirus that we know of. Next, we have the 5-site screen in which Rosanna mutated all 5 positions, and the 4-site screen in which she kept phenylalanine fixed at position 561. I hope you can appreciate that we have now discovered towards the right-hand side of the histogram variants of MXA that are only different from wild-type human MXA at 2 or 3 positions, but are nonetheless tenfold superior to wild-type human MXA at their Togotovirus restriction. And they're actually superior at all different dosages of expression. You can see here that wild-type MXA restriction of Togotovirus goes up as you increase the amount of MXA being expressed, but at the equivalent amounts of expression, the super restrictor MXA variants are significantly better than wild-type MXA. And the reason they're significantly better was actually revealed by co-immunoprecipitation assays by our colleagues at the University of Freiburg, who showed that these variants are able to better bind the nucleoprotein targets and therefore create a more avid interaction that leads to higher restriction. So essentially what we've done is we've taken the evolutionary playbook that we've been given by positive selection to identify variants that are significantly better at binding viral targets and therefore be very specific against that particular virus with increased potency. Our analysis also revealed to us something very interesting about epistasis because we were able to identify single mutations, for example, VFFSS to AFFSS, which is in the backbone of the wild-type human MXA, which is actually a gain of function, whereas the same V to A mutation in the context of the super restrictor that we discovered is actually a loss of function. So this really emphasizes the value of the combinatorial mutagenesis approach because these are probably variants that we would have not stumbled upon if we had simply carried out a single residue mutational analysis of the loop L4 domain. So to summarize what I've shown you so far, we started off with human MXA and showed that the rapidly evolving loop L4 is the primary determinant not just against Togotovirus but also against influenza-like viruses. And now we've taken this combinatorial mutagenesis approach centered on these adaptively evolving residues to identify super restrictor variants that are much more potent against Togotovirus. Of course, we are very interested in understanding whether these variants are also much more effective against influenza-like viruses. That would be a remarkable thing because that would show that you can be a general super restrictor against orthomyxoviruses. In contrast, what we actually found was that there was a significant breadth specificity trade-off where gain of function against Togotovirus in most cases led to a loss of function against influenza-like viruses in general. So we are actually very interested in this breadth specificity trade-off and why it occurs, and actually interested in using the same strategy to identify variants of MXA that might be super restrictors against influenza-like viruses, including viruses that we really care about in terms of their pandemic potential. This is work that's being carried out by Rachel Geiger, a new graduate student who's just joined the lab. So to summarize the work that I've shown you so far, I've shown you that these disordered loop domains and adaptive evolution within these may provide both evolutionary flexibility as well as the adaptive potential for antiviral proteins such as TRIM5 alpha and MXA to keep pace with rapidly changing viruses as part of these Red Queen type interactions. With that, I'd like to thank the people who did the work. I'd especially like to thank my colleague Michael Emmerman, who is a bona fide virologist, and collaborations with him have really been the centerpiece of this work in my lab. I'd like to thank both Rosanna as well as Jeanette for leading the work on MXA as well as TRIM5 alpha. And I'm really excited to engage with you both in the speaker engagement session that immediately follows this talk, as well as in the SMBE business meetings, but also throughout the meeting in the Gather Town that we've actually engineered for you. I hope you will enjoy this meeting, and I hope you enjoyed this talk as well.